you're in trouble. You're going to have to make a hole. If you come in later, make sure you get handouts and hit the sign-in sheet. So we want to circle the coffee table. Anybody else? Oh, yes.
Okay, thank you. I wanted you to know more on the summer about the soil organic matter and the calculation. It being said here that the default depth is 30 centimeter. Depth is usually chosen by the country. I want to know why is it chosen by country when you know that we have trees which roots will go down, other will go just horizontally. Why did you say that it may depends on the type of the forest or the type of the tree, but allow the country to choose that this is my problem? I can't understand. I rather think that it should be good to choose it depending on the type of trees that we can have.
I think that's why they set a default but left it open to the country to decide. Because in some countries, soils are quite shallow. And so a lot of places I work, I sample just to 20 centimeters. And people always ask me, why aren't you going to a meter? Well, I will hand you a shovel. And you can go to a meter. Because I live in a state called the Granite State. And it's all, once you get down much further, you're going to hit granite. So I think they didn't specify a depth because it varies so much, and it may be very important in some systems to go below a meter. And in other systems, maybe you can't get much past 30 or 40 centimeters. So it's largely, they used to say a meter, but now it's largely been left open. But again, if you're doing a RED project or some of these other programs, that might be something you work out with the program funder and settle on that. So there aren't a lot of hard and fast rules in some of this. And sometimes you can only do what you can do. Yeah, I mean, it's just logistically, if I went to a meter, I would make one hole in a plot because I have to use a shovel. So I use a core and I can make 20 holes in a plot, and I feel that I get a better estimate that way. So it's just really tricky. It sort of depends on your situation as to what's going to make the most sense and meet your creation needs. Anyone else? Great.
Regarding the depth of sampling, is it not advisable to get to at least 30 centimeters if you can? Because I think the default that is given by the IPCC is based on the premise that if you just sample, for example, the top 10 centimeters, there is a possibility that it will not give you a fair representation of what may be down. But then, if the soils allow you, you should go to a minimum of 30 centimeters.
I believe that's what they have set 30 as sort of their optional default.
Yeah.
I always tell people, as far as you can go. I tend to hit, personally, a lot of rock right at 22 centimeters, oddly enough. I actually got rid of my 30 centimeter core because I kept breaking it on the rock. So you should always go as deep as it's feasible to go. You want as complete picture as you can. All right, thanks for hanging in there so far. We'll have a coffee break, and we'll come back at 1:45, and then if people don't mind, I'd sort of like to get a little group photo before we move into the all the math stuff. Do a much better job than we can. And also, I think maybe if you tell us your favorite tree species too, just for fun.
Hi everyone, I'm Natalie Wagner. I'm with the Forest Service as well, and I'm with the Ecosystem Services and Markets Program here, and I'll be talking about a little intro to ecosystem services and green ecosystem services. And my favorite tree species is tulip poplar.
That's mine too.
Oh, excellent.
Okay. So I hope everyone had one of the chocolates because they were delicious. Where is this forest? On my travel log, I think this forest is in Maine. So mixed hardwood forest in Maine. So estimation approaches. This is another part of the really big technical stuff that it's important for us to get through. Not maybe the most exciting bit, but this is really, really critical. I talked a little bit earlier about stocks and fluxes. So what I want you to take away from this picture is not necessarily all the things going on, but this is sort of a classic ecological flow chart, and the important thing to realize is, although you'll notice that a lot of the things in the boxes match those pools we talked about, we have boxes and we have arrows. So, carbon estimation techniques. There are two ways we can go about doing this. We can measure stocks. These are the boxes. Okay, so we had a question about this earlier. You measure the amount of carbon in that pool at a point in time. So, you're measuring the boxes, regardless of which box that is. Stock-based approaches to measure what's in the boxes. You can also measure fluxes, the exchanges between these pools and the exchanges between those pools and the atmosphere. So this is measuring the arrows on that diagram. So both of these methods are used for different purposes, and they do complement each other. So flux measurements. This is all we're going to spend on flux measurements. This is where you have those towers, if you've ever heard of tall tower measurements, and they are technically challenging. You need specialized equipment and trained personnel to install, operate, and maintain the tower. They're pretty expensive to install. There's a lot of instrumentation that goes on the tower and a lot of computer support that's needed. Generally, depending, $60,000 to $80,000 to install, and then they do require continuous maintenance. There are some terrain requirements. This is called the eddy flux covariance method, and there's a lot of complicated, fancy math. The exact footprint of the tower is not really possible to determine, and so it's drawing, and so the tower is measuring the exchange of carbon dioxide between the forest and the atmosphere. But there are a lot of sort of rules and things that have to go right. If you get into where your terrain starts to slope more, one of the problems you can have is things don't mix enough, and that CO2 can sneak out the bottom, down the valley, and escape detection. So you cannot put a tower everywhere. It really does need to be a fairly flat, homogeneous site. We have a tower on our home experimental forest, the Bartlett Experimental Forest in New Hampshire. It was put there as a test because it was sort of pushing the edge of what was thought to be suitable terrain. And they are getting data from it, but they said it's definitely, they can see that they're sort of the extent of where it's possible. And it's not the highest quality data. So towers don't work everywhere. It's really good for looking at short-term dynamics, management impacts, that sort of thing over these homogeneous areas. We talked about forests sometimes being sources and sinks, depending on what's happening in terms of perhaps whether it's wet or dry. And this is how we know that in areas where there are flux towers, and so they're getting readings. You can set how often the data are collected, but they can take readings every minute, and then will average over time. Some of them take measurements every few seconds. And so you can see exactly after a rain event, it'll pick up and really start taking up carbon, and during a dry period, it might really slow down and switch to a source. And the flux towers are how they can see this. So it's really good to help improve our understanding of the forest carbon cycle. And there are networks of towers. There's Carbo Europe, there's Ameriflux, there's Ozflux in Australia, Asiaflux, and so people have these, researchers have these towers, and they work together and they share their data. But this is really best suited for research purposes. In terms of operational or project level or country level, this is really not the tool for the job. And this is just to show you one of my colleagues. These are some of his flux measurement towers in Maine. So you can see they're quite tall, they have a lot of instruments on top of them. And yes, there is somebody who has to be trained to go climb up to the top rather frequently to make adjustments and repair equipment. So, measuring the boxes, stalks. This is technically really straightforward. It might be tedious, but it's technically straightforward. Most of the measurements don't require any specialized equipment, so you don't need all those expensive sensors and things like that. So we talked about earlier, you might already have data for some of these pools if you're really lucky. And you can adapt this to a variety of spatial scales. So this will work for a project, this will work for a county, it will work for a province, it will work for a country. This is really, really adaptable. You don't have to worry about what your footprint is and what exactly you're extrapolating. But of course, you're getting that long term, you're getting that net. If your stocks go up over time, you know you're a sink. If they're going down over time, you know you're a source. But you don't have that instant snapshot that you get with the tower. You can't get that immediate response in the short-term dynamics. But we're going to focus on stocks because technically it's a lot easier. We can practice the math right here. We don't need an inexpensive flex tower. This is what you would do anyway for reporting and estimation for MRB. Everyone uses this stock approach. So that's why we're going to focus on this today, but I just wanted you to be aware because you'll hear about flux measurements, so I wanted you to know a little bit about that. And there's some flexibility in choosing the pools to consider. Now, if you're reporting or you're using the IPCC methods and you're reporting to meet obligations under the Framework Convention, you do have to report all the pools. If you're doing projects and some of these other purposes, you may have some flexibility in which pools to consider, and that would be part of the program documentation or something you might work out with the project funder. But with a tower, it's looking at that total exchange, and so you have to go and do a lot of backup measurements to break that out, and you can't leave out anything. But here, you could choose your pools depending on your purposes. So this is just basically the IPCC says, if we measure the boxes at 2 different points in time, we consider that to be equivalent to a flux. So it's a little snazzier to measure the arrows. Everyone loves flux towers, but it's a lot more practical to measure the boxes. So we're going to focus mostly, although I said I do a lot with soils, we're going to focus on the live biomass carbon pool today. What are the, I'm going to save my last door prize because I'm still hoping there's going to be a haiku that you're all going to think about your forest carbon haikus during lunch. But I'm not kidding. I want to hear them. What are the 2 biggest carbon pools? Anyone know? Of all the carbon pools we talked about, doesn't matter where you are in the world, 2 of them are the biggest, although which one is the largest and the second largest varies depending on where you are in the world. What's one of them? Anyone? Yes?
Oceans.
Sorry?
Ocean.
Okay, we're going to talk about terrestrial. Sorry, my bad. Terrestrial carbon. You're right, oceans are a huge one. In terms of forests, what are the 2 biggest carbon pools in a forest? We know this, come on.
Components, you mean?
Sorry?
The components? The foliage components of the state, something like that. Is it what you mean?
I can't hear you at all.
Foliage, from the foliage of the state, the components?
The above ground biomass, yes. The trees, what's the other one?
Soil.
Yes, soils. Now, depending as you move from south to north, which one of those, so generally the closer to the equator, your live biomass is the biggest one. And as you move up toward, or down, I guess, depending on where you are, toward the poles, your soils become the larger pool, generally. So even though soils are a hugely important pool, we're not going to spend a lot of time on them because in the absence of major disturbance, they change pretty slowly. That's why I was talking about the problem of change detection, because it's quite slow against a large background. But with live biomass, we all know you can get large changes fairly quickly. And this is the one we can influence the most with our management. And certainly this is what we're really focused on if we're talking about restoring forests, putting forests back where they used to be, or taking non-forest and transferring it to forest. So that's the pool we're going to spend the rest of our day on. And there are 2 main ways that you can go about doing this. So one I always kind of call the volume-based approach. So foresters are really interested in wood volume because that's the product. So you may often have these data. And I know FAO collects them, and there's just a lot of different volume estimates out there. So what you can do is take merchantable volume And then you use what's often called a biomass expansion factor. The current guidance calls it a conversion and expansion factor. It's an all-in-one factor. And you convert that to total volume and dry biomass, and then to carbon, using this factor. The IPCC guidance does have default biomass factors, and those of you who stay for the exercise, we will work using them. There are published factors also available, and those factors may not be complete. You may have to also multiply by wood density. So there are two types of factors. The one we use today are a one-stop shop. The other way, the way I most frequently work, is the biomass type approach. So I'm going to calculate stem by stem. And I have a set of equations I like to use that are nationally consistent. And so what I have are tree records of all the numbers of trees and their diameter. And from that I can apply the correct equation and calculate the biomass and then convert it to carbon. The equations for biomass, this is where you can run into trouble depending on where you are in the world. They are regionally specific, they're often species specific, and diameter range is important to consider too. If the equation was developed for a fairly small range of diameters and you're applying it to trees that are very much larger, that is a very large source of error. So choosing the correct equations can be challenging. In equations, somebody mentioned that they were trying to use analogs, that they didn't have equations appropriate to their species, and that absolutely is a problem. And in that case, you may be better off using the IPCC default biomass expansion factors and taking that approach. There may be less error than applying an equation that's not a good fit. You can develop these equations. It is fairly expensive because to develop the equations, what you do is find a bunch of trees of your species of interest, a lot of different diameter ranges, so from small to fairly good sized, and numbers of each of them. You can't just do one. You chop them down, you chop them into bits, you haul them back to the lab, shove them in an oven, and then dry them and weigh them. So then you know the amount of biomass of the tree and the height of the tree and the diameter, and you construct an equation. But you can see that's rather an awful lot of work. And the equations have a number of mathematical forms. There's no one way to do it, but it's not something you want to do unless you have to. But there are cases, I had a phone call from someone, they wanted to do citrus in Mexico, citrus groves. Well, there just wasn't anything that was really going to work, and they actually did get funding to work with the university to go cut down a bunch of trees and make the equations. So this biomass expansion factor, this does require less data, so you just need that merchantable volume. It's an aggregated approach, so it's really quick and easy to apply. Again, you might need wood density. It just depends on your factor. If you're using the IPCC ones, you're fine. If you have some locally developed ones, you may or may not need to use wood density. Because this is based on merchantable volume, it will underestimate the carbon in the smaller stems. The good news is, small stems really don't have that much carbon in them. And I've experimented with this. I had tree records and I dropped out everything under 3 inches. Every tree that was under 3 inches on that plot. And my total carbon estimate hardly budged at all. So this isn't such a big worry. And again, this is an aggregated approach, so when you don't have that individual tree data, you have to go this way. You have no choice if you don't have diameter. And if you have inappropriate equations and you just feel they're not a good fit, you might be better off going this way as well. And some of these, the IPCC factors now, actually the equation, when we look at it later, does already include adding the below ground biomass. If you're using a factor that doesn't, you just need to compute that and add it in. But it's easy, very simple to apply. This is the other way, the way I often work. Most equations will often require height. Will require height. You can't calculate height from diameter. I use equations that don't actually require height, which is nice. These are more accurate for smaller stems, but again, that's not such a big worry. I wouldn't let that keep you up at night. Usually these are for above ground biomass only. There may be separate equations for the roots that were developed as part of the study, or you can use the published root to shoot ratios from IPCC and calculate that. Again, 20% is not a bad rule of thumb. There are published equations that are often available, but as we mentioned, they may not be available for your species, or they might have been developed on a really dry site, or your trees are much larger than those for which the equations were developed. Equation choice is one of those things you need to be careful about, and you need to document carefully the reasons you selected those equations. So there's a lot of room for error there.
Okay, this is a question. Going to your previous slide, you said for this is the equation, the expansion factor, it underestimates small trees. And this is more for the small stem, the allometric equation is more. But for one of the REDD plus activities is restoration, right? So if a country has just started planting, started restoring, so would they not need this more of allometric equation method, so that they can report like after only 2 years or 3 years rather than wait for 10 years?
Right, that's a really good question. There's not a lot